Hi class, we're going to dive into chapter 14. Now, depending on what class you have me for, I make all of these videos for my A&P 1 classes. My Hartford Community College students, you guys are in chapter 14 for this. CCBC uses a different book. You guys are in chapter 16 for this. So this is the chapter on the autonomic nervous system, just to make it all clear to you. Even though it says chapter 14, I prefer this version of the information. It's the same information no matter which book you're in. It's just this book calls it chapter 14, the other book calls it chapter 16. It's the same stuff. Just a quick overview. The somatic nervous system is our conscious and subconscious control over our skeletal muscles. The pathways include upper and lower motor neurons. Typically, the lower motor neuron is controlled by like a reflex. The upper motor neuron has a cell body up in the brain or the primary cortex. And so that's somatic stuff. We're going to get into autonomic stuff. This is our visceral, mostly outside of our awareness, not in our conscious control or regulation. The motor neurons carry out information that comes from the hypothalamus and all of the other autonomic centers of our brain. They're basically comparable to the upper motor neurons of the somatic motor system or somatic nervous system. Our autonomic centers basically have a series of two neurons. Instead of having an upper and lower motor neuron like in the somatic nervous system, we have a preganglionic neuron and a ganglionic neuron. It's the same idea, that's just what they're called. So, preganglionic neurons, these have cell bodies within the brainstem or spinal cord. They're part of a visceral reflex arc, and most activities that they direct are reflex type responses. Like, you got to undergo peristalsis, or it's time to regulate your, little sphincter muscle at your stomach or whatever. All those things, visceral reflexes, you don't really need to be aware of them. They leave the central nervous system, they synapse with a ganglionic neuron. So the ganglionic neurons have cell bodies in the peripheral autonomic ganglia. There's hundreds of thousands of ganglionic neurons. They innervate all of our visceral effectors, cardiac muscle, smooth muscle, glands, and adipose tissue. So here you see what I mean. The visceral nuclei within the hypothalamus send signals down out the autonomic nuclei of the brainstem or down through the spinal cord to a preganglionic neuron. The preganglionic neuron has a cell body inside the central nervous system, and then it synapses somewhere with a ganglionic neuron, which goes to a peripheral visceral effector. So you can pause the video here, go back and review these 3 questions. Let's talk about the major divisions of our autonomic nervous system, the sympathetic and parasympathetic. They have opposing effects. They can work independently or they can work together. Most organs and structures are innervated by both sympathetic and parasympathetic nerve fibers, but some are only innervated by one or the other. Both divisions influence a third division of our autonomic nervous system called the enteric nervous system, or ENS. So let's talk about our sympathetic division. In general, this is all of our emergency, stress, exertion stuff, fight or flight, right? It's also called the thoracolumnar division, but we can just call it sympathetic division. The axons emerge from cell bodies on the thoracic and lumbar segments of the spinal cord, hence thoracolumnar. The axons innervate ganglia that are relatively close to the spinal cord as well. So, in parasympathetic, these are called the craniosacral division. This is our resting conditions, rest and digest. The axons emerge either from the brainstem or from the sacral segments of the spinal cord. These innervate ganglia that are very close to or within their target organs. So here you can see the difference. Sympathetic, our ganglionic neurons are coming out of the spinal cord through the thoracic and lumbar segments, synapsing with the ganglionic neuron in close proximity to the central nervous system through something called the sympathetic chain ganglia. There's some other ones that we'll talk about. The parasympathetic division, includes the cranial nerves, some of the cranial nerves, and some sacral nerves. So let's talk about that ENS, enteric nervous system. The enteric nervous system is an an extensive network of neurons that's within the digestive tract. This is influenced by sympathetic and parasympathetic divisions. It initiates and coordinates all the visceral reflexes of the digestive system. It uses the same neurotransmitters that are found in the brain, and we'll talk about it later when we get to the digestive system next semester. There it is. Here are our autonomic plexuses and ganglia. So you can see these sympathetic chain ganglia along the sides, and some of the other inferior mesenteric ganglia and celiac ganglia, etc. You can pause the video here, go back, and review this little section. Let's talk about the organization of that sympathetic division. So the preganglionic neurons of our sympathetic division synapse on the ganglionic neurons, which are within the sympathetic chain, the collateral, or the adrenal medulla. So the sympathetic chain is a series of interconnected ganglia that are on either side of the vertebral column. The collateral ganglia are located within the thoracic and abdominal pelvic cavities. These are the celiac, superior, inferior mesenteric ganglia, or the adrenal medulla. Adrenal medulla is on the adrenal glands. So sympathetic division has preganglionic fibers that are relatively short. They're just coming out of the spinal cord, the thoracolumnar area, and then the postganglionic fibers are synapsing within those ganglia, like the sympathetic chain ganglia, for example, and then going relatively far to their peripheral effector. So again, lateral gray horns of spinal segments, thoracic segments of the spinal cord, either synapsing to their ganglionic fibers at the sympathetic chain, collateral, or adrenal medulla, and those ganglionic fibers are then going to the target organs, visceral effectors, etc. All right, let's talk about some responses to increased sympathetic activity. This is fight or flight, heightened mental awareness, increased metabolic activity, decreased digestive urinary functions, decreased reproductive functions, mobilization of energy reserves, dilation of respiratory passageways, pupils, etc., increased heart rate, increased blood pressure, activation of sweat glands. This is the, oh my gosh, the plane's about to take off and I'm nervous that we're going to crash and die type of feeling. Parasympathetic division is very different. The preganglionic fibers synapse on 6 to 8 ganglionic neurons, and these are located in what we call the terminal ganglia or the intramural ganglia. Terminal ganglia are located near a target organ, very, very close to it. They're usually unpaired. These are the ones that are associated with the cranial nerve, or the intramural ganglion, which are embedded in the tissues of the target organ itself. These are interconnected masses of ganglion cells. So again, in parasympathetic, we're talking about cranial nerves 3, 7, 9, and 10, all going to their target organs, or the pelvic nerves, again, going to their target organs through the intramural ganglia. Let's talk about some responses to parasympathetic activity. This is rest and digest, decreased metabolic rate, decreased heart rate, blood pressure, increased secretion of salivary and digestive juices, increased motility of blood to the digestive tract, and stimulation of urination and defecation. So then you might say, well, I know that the sympathetic nervous system is fight or flight, and I know that the parasympathetic system is rest and digest, but right before a test, I have to pee. Or right when I get nervous, I have to pee. And that's because your parasympathetic is relaxing all of those sphincter muscles. So you might get that sudden urge to go to the bathroom once you start to get nervous all of a sudden, because your parasympathetic nervous system isn't pushing the urine out or pushing the feces out, but those sphincter muscles are relaxing because you're focusing your attention somewhere else in your body. Which is why sometimes you have to pee a lot or go number 2 a lot before something major that you're stressed about. You can pause the video here, now that you did that little tidbit, and answer these 3 questions. Let's talk about how our sympathetic division is innervated. Every spinal nerve has a gray ramus carrying a sympathetic postganglionic fiber. It distributes them to the body walls and the limbs. The head and neck, the postganglionic sympathetic fibers from the superior cervical sympathetic ganglia are innervated by the cranial nerves 3, 9, 7, and 10. And there are groups called splanchnic nerves that supply the viscera, the cardiopulmonary, abdominal pelvic, and pelvic fibers. All come from the splanchnic nerves. There's the splenic nerves. And then you also have the parasympathetic innervation, most of which is coming from the vagus nerve. 75% of all the parasympathetic output is coming from the vagus nerve, cranial nerve 10. Lots of branches going through the thoracic and abdominal pelvic cavities, the preganglionic fibers in the sacral spinal cord segments carry the sacral parasympathetic output. So they don't join the ventral roots of the spinal nerves, they form the pelvic nerves. The pelvic nerves innervate the intramural ganglia within the kidneys, the bladder, the terminal portions of the intestine, and the sex organs. There you see the pelvic nerves coming off the sacral spinal nerves, and then of course we have our cranial nerves 3, 7, 9, and 10. You can pause the video here, go back and answer these 3 questions. Moving on, let's talk about special neurotransmitters. Neurotransmitters released in the sympathetic nervous system are mostly androgenic. Androgenic means they are releasing epinephrine or norepinephrine. They are located on the plasma membranes of our target cells, and there's 2 types, alpha and beta. So the neurotransmitters binding to androgenic receptors are epinephrine and norepinephrine. Norepinephrine stimulates the alpha receptors, and epinephrine stimulates both alpha and beta receptors. Localized sympathetic activity typically involves a release of norepinephrine at the synaptic terminals and primarily affects the alpha receptors near those terminals. The effects last for just a few seconds. Generalized sympathetic activity involves epinephrine and norepinephrine being released by the adrenal medulla, affecting both alpha and beta receptors. 3 times as much epinephrine is released, so it has a greater effect on those beta receptors. So the effects are more long-lasting, can last up to 30 seconds, and metabolic efforts can persist for several minutes after being released. So those alpha receptors, as I said, generally stimulated by epinephrine and norepinephrine, stimulate what we call a G protein. The G protein is going to trigger this kind of cascade pathway inside the cell. So your alpha-1 receptors are excitatory and alpha-2 receptors are inhibitory. So here you see what I mean. Epinephrine comes in, joins to an alpha receptor, our G protein is now activated. When that G protein is activated, it can reduce C-camp levels if it's an alpha-2 receptor. If it's an alpha-1 receptor, our C-camp levels are going to increase, which is going to release calcium from the ER, which is going to stimulate either gland secretion or smooth muscle contraction. If the C-amp levels are, it's cyclic AMP. If cyclic AMP levels are decreased by being inhibited by an alpha-2, then the cell is going to be inhibited and those glands will not secrete and the muscles will not contract. So beta receptors are generally stimulated by epinephrine. They're located in many organs like skeletal muscles, lungs, heart, and liver, stimulating the beta receptors, and a G protein is going to trigger metabolic activity in the target cell. And there's 3 types of beta receptors, beta 1, beta 2, beta 3. Here you see what I mean. Our beta receptor G protein's inactive. Once epinephrine binds, then we have an activated G protein, activated adenylate cyclase to create cyclic AMP. If it's a beta 1 receptor, cardiac muscle is going to be excited, tissue metabolism is excited. Beta 2 receptor is going to relax smooth muscle in the respiratory passageways, blood vessels, etc. Beta 3 is going to release the fatty acids from the adipose to mobilize metabolic reserves into our other tissues. There's also different neurotransmitters in the parasympathetic division. So sympathetic uses epinephrine and norepinephrine with the alpha-1, alpha-2, beta 1, 2, 3 receptors. Parasympathetic uses acetylcholine. These are called cholinergic receptors, and there's 2 types. There's nicotinic and muscarinic. Nicotinic receptors are found in all postganglionic neurons. They're found on the adrenal medullae, and they're found in all neuromuscular junctions of skeletal muscle cells. They're always excitatory, and they're stimulated by nicotine, a powerful toxin in our body. Here you see what I mean. ACH binds, sodium channels open, sodium rushes in. That sodium channel is a nicotinic receptor. Cholinergic receptors that are muscarinic have G proteins that are activated in their pathways. So they occur at a cholinergic neuromuscular or neuroglandular junction in the parasympathetic, as well as a few cholinergic junctions in the sympathetic division. Activation is going to be longer lasting. The response can be excitatory or inhibitory in a muscarinic, and it's stimulated by muscarine, which is a toxin produced by poisonous mushrooms. So again, here You have your muscarinic receptor taking acetylcholine, activating G protein. It can activate or inactivate special enzymes and have metabolic effects. You can pause the video here, go back and answer these 3 questions. Let's talk about some differences between sympathetic and parasympathetic. Overall changes in activities by releasing norepinephrine at the peripheral synapses or distributing norepinephrine and epinephrine throughout the body. Can also have long-term local effects by activating specific effectors. Sympathetic activation will typically occur in a crisis. This is, again, our fight or flight. Sympathetic centers in our hypothalamus control the sympathetic activation, and the effects are not limited to peripheral tissues. It can also affect central nervous tissue function as well. This is the overall summary of sympathetic activation. All of the things that occur when sympathetic activation is occurring in your body. Here's the breakdown of the structure. So again, in the central nervous system, you have your preganglionic neuron, synapsing at the adrenal medulla, sympathetic chain, or collateral ganglia. This is adrenal medulla. So here we have our sympathetic ganglion, ganglionic neurons, and postganglionic fibers going into a target tissue or secreting norepinephrine, epinephrine into the bloodstream to go to target tissues. This is just breaking down the overall characteristics of the fibers in the sympathetic division. Let's talk about the parasympathetic division. Under normal conditions, the entire system is either controlled nor activated as a whole. So it's just parts that are activated continuously. So the activities can be reflex responses to conditions within specific structures or regions. It's not going to affect your body-wide. It's just going to affect different parts. Sympathetic activation is widespread, all hands on deck. Parasympathetic is more localized and individualized. So the effects center on relaxation, food processing, and energy absorption. Rest and digest. That's all what parasympathetic is. It's also called the anabolic division because the stimulation of parasympathetic leads to increased nutrients in the blood, causes these nutrients, or cells need these nutrients for growth, development, and excess can be stored in the lipids or glycogen reserves for energy later. This summarizes all of the parasympathetic effects. Here you see the parasympathetic structure, preganglionic neuron in the central nervous system, either from a cranial nerve or sacral nerves, preganglionic fiber going out to intramural ganglia or our target tissues here, parasympathetic ganglion, just synapsing with our target tissue through the postganglionic fiber. Here's our general characteristics of our parasympathetic division. You can pause the video here and answer these 3 questions. We have some more autonomic regulations and control mechanisms, some outcomes that are already listed for you in your chapter folder or weekly folder. Let's talk about how our autonomic nervous system maintains homeostasis. It's vital for physiological processes to continue with or without consciousness. You need your autonomic nervous system. People can survive for decades in a coma just because of their autonomic nervous system. The autonomic nervous system adjusts activities for digestion, cardiovascular, respiratory, reproductive without any input from the conscious mind. So any output from the autonomic nervous system is going to impact virtually every system in the body. So some type of stimulus being sensed by our general sensory receptors, sensory pathways, communicating that information back to sensory processing, all of this going through motor pathways, either somatic or visceral. Somatic is our skeletal muscles, visceral is our autonomic nervous system with all of our smooth muscles, glands, cardiac muscle, and adipocytes. I believe we've seen this one before. You can pause the video here, go back and review that nice little flow chart. Most of our organs are dual innervated. In other words, they have innervation from sympathetic and parasympathetic nervous systems. This provides an autonomic tone. It's a continuous level of spontaneous activity. So it's maintained in the absence of stimuli, kind of like skeletal muscle tone, how they will remain slightly contracted and have tension on them even at rest. Think of your muscles like that also. Your smooth muscles, your glands, your adipose, all of those organs are still maintaining a degree of activation even if they're not being activated. With dual innervation, they're receiving instructions from both sympathetic and parasympathetic. This is occurring in most of our vital organs. The effects can be complementary or opposite and opposing. In organs with only sympathetic innervation, the responses can vary depending on the type of receptor. So here you see, again, those alpha 1, beta 1, beta 2, beta 3 receptors are playing a role in whether or not things are inhibited or excited. And these are some structures that are dual innervated, like our lacrimal glands, or our sweat glands, or skin, blood vessels in our skin, veins, heart, etc. All dual innervated, and then the effects from the parasympathetic and sympathetic. Do you have to have this list memorized? No, but if I asked you, what would be the effect of dual innervation on the salivary glands? Well, salivary is digestive. So with parasympathetic, that's increasing secretion. With sympathetic, that would be decreasing secretion. That's all you need to know. Just a general overview. Dual innervation can have effects on the heart. The heart's composed of cardiac muscle tissues. Contractions are triggered by pacemaker cells. The autonomic nervous system has opposing effects on those pacemaker cells. ACH is released by parasympathetic. Norepinephrine is released by the sympathetic. So small amounts of both neurotransmitters are continuously released. Under resting conditions, the parasympathetic effects dominate in your heart. So dual innervation at your heart. At rest, both ANS divisions are active, but at low levels, that parasympathetic dominates. With increased parasympathetic input, we're lowering our heart rate. If the parasympathetic is inhibited, it's kind of taking the brakes off. So our heart rate goes up, and now we get sympathetic stimulation on top of the fact that parasympathetic has been inhibited. So now we're going to get our heart rate up to maximum levels. You can pause the video here, go back and answer these 3 questions. Let's move on to visceral reflexes. Visceral reflexes provide automatic motor responses that can be modified or facilitated or inhibited by those higher centers of the brain, specifically the hypothalamus, just like our spinal reflexes. So they're polysynaptic, unlike most of our other spinal reflexes, which are monosynaptic. Each reflex consists of a receptor, sensory neuron, processing center, 2 or more visceral motor neurons. There are short reflexes. Short reflexes bypass the central nervous system entirely. The cell bodies of the interneurons are located within the autonomic ganglia, and those interneurons just synapse back with the ganglionic neurons. The motor commands can be distributed to the effectors by the postganglionic fibers. This controls very simple motor responses with very localized effects, one activity within a target organ. So this is very much how our enteric nervous system operates. You have some type of stimulus, maybe food just arrived in your small intestine, and your brain doesn't really need to know that because you already know that you ate. You consciously know that you ate food. You consciously felt the distension or stretching in your stomach, but you don't need to know that the food has now passed from your stomach into the small intestine. So this is a short reflex. This is just going to occur right in that local area, saying, okay, now that the food's in the duodenum, now it's in the small intestine, now we need to add some digestive juices in there. That's all a short reflex is. A long reflex is the same thing, except the sensory neurons are going to take that information up to the central nervous system. So the dorsal roots of the spinal nerves are going to take that information up to the brain, or within the sensory branches of cranial nerves, or within the autonomic nerves that are within the visceral effector. So interneurons are going to process that information in the central nervous system and then carry out motor commands to visceral effectors. It's predominant over short reflexes and coordinate responses between multiple organ systems. So again, some type of stimulus, the long reflex carried out to a peripheral effector. Here's some common visceral reflexes: cardiocirculatory, vasomotor, pupillary, ejaculation, defecation, urination. These are all reflexes. Coughing, baroreceptor, swallowing. Again, all reflexes, all reflexes that have a stimulus and a specific response. So let's talk about how these visceral reflexes are sensed. We need a receptor. Let's talk about them. Interoreceptors are collecting visceral sensory information, monitoring visceral tissues, primarily in the thoracic and abdominal pelvic cavities. These are nociceptors, thermoreceptors, tactile receptors, baroreceptors, and chemoreceptors. We have solitary nuclei, which is the main processing center for all of the interoceptors. That's done at a subconscious level. It's nuclei in the spinal cord. The solitary nuclei is in the brainstem. So major processing and sorting for all the visceral sensory information is done within the solitary nucleus of the brainstem. Extensive connections with the cardiovascular respiratory centers as well as the reticular formation are all going through this nucleus so that they're all communicating together, and the information seldom goes up to the cortex for awareness. So all of that internal stuff that's going on is mostly relayed right at the brainstem instead of going up to our conscious awareness. Thank goodness. Otherwise, our brains would just be really full of extra stuff that we didn't really need to know. Here are 3 questions. You can pause the video, go back and review. Let's talk about the role of baroreceptors. Baroreceptors are special stretch receptors to monitor changes in pressure. The changes in pressure will distort any of the dendritic branches and alter the rate of action potential generation. So they're kind of like mechanically gated channels. The free nerve endings branching into the elastic tissues within hollow organs, blood vessels, tubes like the respiratory, digestive, and urinary tracts, all to say, hey, our bladder's full, we need to go to the bathroom, or hey, our stomach's full, we can be done eating now, right? Baroreceptors also monitor blood pressure within the walls of our major vessels. They're located in the carotid artery, the carotid sinus, the aorta, at the aortic sinus, and information plays a major role in regulating our cardiac function and adjusting blood flow to various tissues. There's also monitoring the degree of lung expansion. It's relayed to the respiratory rhythmicity centers of the medulla oblongata, which sets the pace for respiration. It triggers the visceral reflex in the digestive and urinary tracts, like urination. There you see some of the baroreceptors and where they are in the body. And we also have chemoreceptors. Chemoreceptors are specialized neurons that detect small changes in concentrations of specific chemicals or compounds. They play that role in reflexive control of respiration and cardiovascular function. So there's some specialized locations of chemoreceptors, the medulla oblongata and other places of the brain where there's a break in the blood-brain barrier to monitor pH, to monitor the partial pressure of carbon dioxide in the CSF, very important. The carotid bodies located in the internal carotid arteries, again, pH, partial pressure of oxygen and carbon dioxide of the arterial blood, and in the aortic bodies, between the branches of the aortic arch, monitoring pH, partial pressure of carbon dioxide and oxygen in the arterial blood as well. You can pause the video here, go back and review. And our autonomic nervous system can be controlled, again, through basic visceral reflexes or more complex reflexes. The basic visceral reflexes are controlled by the nuclei of the brain stem and spinal cord. These are relatively rapid, automatic responses to various stimuli. The more complex reflexes are coordinated by processing centers within the medulla oblongata. So these are our cardiovascular, respiratory centers, the centers and nuclei involved in things like salivating, swallowing, digesting, peristalsis, urinary function, all more complex reflexes. So the centers within the medulla oblongata are all regulated by the hypothalamus. So all of that control comes down from the top, so to speak. The hypothalamus acts as this headquarters of the sympathetic and parasympathetic division, interacting with all the other parts of the brain, including the limbic system and the thalamus and the cerebral cortex, tying it all together in the autonomic function. So heart rate acceleration with anger, right? You get angry and all of a sudden your heart's beating out of your chest. It provides continual feedback between those higher centers of the brain, the hypothalamus and the brainstem. So here you see all of this interaction, right? Cerebral cortex communicating at a subconscious level, limbic system with its emotion, memory, and behaviors. Thalamus is always relaying that sensory information. Hypothalamus is parasympathetic and sympathetic headquarters, regulating respiration and heart rate in the pons and medulla oblongata, signaling all those reflexes out through the spinal cord and sacral spinal cord with our vasomotor, swallowing, coughing, respiratory, etc. reflexes. So, overall patterns of organization. Both the somatic nervous system and the autonomic nervous system have parallel organization. So they're integrated at many levels. Sensory pathways carry information distributed to both the somatic nervous system and the autonomic nervous system. So it's an integrated, compatible reflex. Higher levels of integration involve the brainstem. Both systems can be influenced by higher brain centers. So it's the same basic setup. It's just a different pathway of nerves and hormones influencing. So here you see the overall difference between some of our somatic reflexes and some of our autonomic reflexes, like our long and short reflexes, but it's basically the same. We have the same levels of control happening, same levels of intervention, all of the same parts of the brain are involved for the most part. It's just a different setup. This is the last slide of the video for this chapter on the autonomic nervous system. You can pause the video, answer these 3 questions. You can post questions on our discussion board for this chapter. You can email me questions. Hopefully we'll get a chance to talk in class or in my office hours. So let me know what questions you had on this chapter. It was pretty straightforward, just talking about the difference between sympathetic and parasympathetic divisions of our autonomic nervous system. Have a good day.